All info from this video is from my genetics textbook and PowerPoint, and then all images are from Google Images. So, transcription is the process of formation of mRNA from the non-coding strand, which is the 3' to 5' strand, and the mRNA formed is the exact copy of the coding strand, which is the 5' to 3' strand, except thymine is replaced with uracil. So when we transcribe the coding strand, we get AUG, UGG, AUU, ACU, UGC, UGA, CCA, and UGC. And then you have to translate that. And that is the process of formation of the proteins, which are the amino acids, from the codons present. And the tRNA is involved, which consists of anticodons with the codons of the mRNA. So the translation, you use the amino acid chart right here, and you code, so like AUG would be AUG, and you get MET. So we have MET, TRP, ILE, THR, and CYS. And then we have a stop codon, which would be UGA, and you'll code that like this, U, G, and then A. So it's a stop codon. The first mutation that I chose was the Missense mutation, and that is where the single nucleotide changes, resulting in a codon that codes for a different amino acid. So I changed ATT to ACT, which is a transition mutation, because transition means it interchanges of the 2-ring purines, which is A and G, or the 1-ring pyrimidines, which is C and T. So this is C and T. And it's like the speed limit, where you get one speed limit and then it changes to another speed limit. So we have AUU, which codes to ILE, and then ACU, which then codes to THR. Then I had the nonsense mutation, which it's just changing the DNA that causes a protein to terminate the translation earlier than expected. So it's a premature stop codon, like these traffic lights. You're driving and then your light's green and then all of a sudden the light's red, so then you have to stop. You can't go any further. So I changed TGG to TGC, which is a transversion nonsense mutation, because the transversion means that it's an interchange of the purines for pyrimidines in exchange of a 1-ring to a 2-ring structure, a 2-ring to a 1-ring. So we get UGG, which is TRP, to UGC, which is a stop codon. Then I have a silent mutation, which is a change in the sequence of the nucleotide that causes a different codon, but it doesn't cause a different amino acid because it codes for the same one. So we have 2 different speed limit signs, but they're both 5 miles per hour. So it doesn't change your speed, it just, the look of it changes. So that would be transversion, because it's T to A, not T to C. Silent mutation, ACU equals THR, and ACA equals THR. And then the read-through mutation changes a normal stop codon into an encoding codon, causing extension of the polypeptide change. So it goes from a stop codon to a go codon. So we have a red traffic light, we're driving, and we're stopped, and then all of a sudden it turns green, so then we can actually continue driving. So I changed TGA to TGG, and this is transition, and I got UGA, which is a stop codon, to a UGG, which is a TRP codon, which extends the polypeptide change.